started. Let's get started again. We'll get started. We'll start the debrief. So we've had 4 working groups that have met, and I think we've had some really interesting discussions. I was able to attend some of the discussions briefly. I think we have some really good recommendations and suggestions. So we'll go on the order of the working groups there on the screen. We'll try to maybe stick to around 15-20 minutes, per group. That's an upper bound. If you guys want to, take less time, that's totally fine. We would like to have everyone be as engaged as possible. So, please feel free to ask questions and, make it as interactive as possible. I'm not going to moderate the working groups. I think, the moderators or the speakers will do the moderation.
I have some slides, but I need to switch to VGA. So I have no idea how to do that. Could you switch to VGA? Yeah. I plugged it in too, but it doesn't. Maybe I should just. Yeah. Could I switch? Could you switch to video?
It's already. That's it.
Yeah. It doesn't recognize it. Oh, I see. All right. I had to unplug the other computer. So I'm happy to report back on the hard work that our working group did, the working group on liquid biopsies. So first of all, to get everybody on the same page, liquid biopsies are really just measurements, often genomic measurements of nucleic acids that one can isolate from the blood, and CTCs. I've been asked to not forget CTCs, and I'll try to remind everybody of CTCs. But nucleic acids that can be isolated from blood. Often we think about cell-free DNA first. These molecules are very abundant. They exist in genome equivalents of about 1,000 to 10,000 genome equivalents. And they're very responsive to DNA sequencing technologies. And several applications have been pursued, including non-invasive prenatal testing. Was the first liquid biopsy or first application of liquid biopsies that made it to the market. Was first described in 2008, became commercially available in 2010, and is now the most commonly used genetic test out there. It's more often used than the 23andMe genotyping arrays. So an enormous success. Excuse me? Oh, excuse me. Then cancer screening is another application, and as you'll see, a lot of the people that were involved in the working group focus on cancer, and we've learned also today that many of these cancer cell-free DNA tests are already in use at Weill Cornell Medicine, are being ordered at Weill Cornell Medicine. Solid organ transplant monitoring is something that I've worked on a lot, is also commercially available, and is having some impact on clinical care and also infectious disease diagnosis, as I mentioned this morning. Many of the participants in the working group, not all, we had a big group we had a lot of people in the room, but about eight of us have active research in liquid biopsies. 6 out of 8 work on cancer, circulating tumor cells, or cell-free DNA, and use these assays to look at response to therapy, develop prognostic markers. There was also some nice animal model work and animal investigation of cell-free DNA in canine cancers. There was a project on chronic fatigue syndrome, and the use of the question was whether cell-free DNA could be useful in that context too. And then my group works again on transplant complications and infections. There are many opportunities for liquid biopsies, in particular for this type of cross-campus interaction. First of all, often these samples are non-invasive samples. They're often previously biobanked. So my lab has done a lot of work with the transplant unit here And we had the opportunity to simply just go to collaborators and ask them to send us retrospective urine or plasma samples. So often these samples already exist. There's no need to design a de novo study, and that enables one to hit the ground running. The samples can be stored for a long time. In the case of plasma cell-free DNA, you can, the cell-free DNA has its integrity for more than a decade. So it's easy to design and perform longitudinal studies. Setting up IRBs is also of interest to note. Often straightforward, not only because the samples are non-invasive, but also because they're often archived, already available. And so for example, researchers on the Cornell campus, if you want to interact with physicians that have stored blood samples or urine samples or other biofluids, and you want to investigate liquid biopsies, often the only thing you need is a request for exemption from further review, which is often granted in about a week or so. Another great opportunity, I think, for liquid biopsies is that the measurement itself, the measurement of human health itself, is also the translation. So once you can establish the utility of some type of measurement, that is also the product, right? You're not looking for specific targets for therapeutics. The measurement itself is the end product, and that means that explains also why many of these tests have found access to clinical care so quickly, have been able to impact clinical care so quickly. So often within 2-3 years, concepts that are developed in the lab can be brought to the market and can be applied and can impact clinical care. And so that is certainly inspiring. There are few challenges that were brought up. We had a group of very positive thinkers, so we've largely talked about opportunities. One of the challenges that came up is bioinformatics, often complex. Often new data analysis approaches are needed, and there's not enough interaction between different groups that work on these concepts to share principles and so on. And it can be expensive, including reagents can be expensive. For example, the tubes, the best possible tubes to store blood in if you want to develop a liquid biopsy based on blood is something called a Streck tube, which costs about $10 per sample, which can actually be a hurdle for clinical collaborators if you ask them to collect many hundreds of samples for you. So that is one challenge that came up. There were lots of suggestions for concrete research directions that came out of our discussion. It was suggested that we look for new fluids, lung fluids, peritoneal fluid, urine, that we spend more time thinking about animal models. There's a lot of opportunity for genomic medicine researchers, engineers, both in Cornell and here, to develop novel measurements, novel measurement principles. For example, to interrogate novel epigenetic marks, to create novel measurements of these epigenetic marks, and so on. There's a lot of opportunity for new applications. Too much of the focus is on cancer. There's likely lots of opportunities for liquid biopsies outside of cancer. For the engineers in Ithaca, there's a lot of opportunity likely for the development of devices and interfaces to create point-of-care tests, for example, paper-based tests and so on, that take advantage of these concepts of liquid biopsies, and those may be overlooked. Liquid biopsies are very DNA-centric. CTCs are often not mentioned or not enough mentioned. And also it was brought up, why not focus on RNA? Why not look at protein technologies, metabolites, and so on? Those are great possible research directions. There were several actionable items that came out of our discussion. The first one, I think, could be useful for all the working groups, and that is to have some type of shared computational infrastructure. This has been brought up before. This is not an entirely new idea, but space is cheaper in Ithaca, so why not build out sort of a shared, large, high-performance computing facility on one campus that everybody has access to, which would then enable great opportunities for collaboration, certainly on topics of data science. There should be a bigger push to integrate technologies. At meetings like these, we sometimes discover that we're collaborating with the same physicians, looking at similar questions, using different technologies, and so there's a big opportunity to integrate these type of technologies and measurements more that we should not overlook. There could be better support for cross-campus IRB applications. Often it can be challenging to move something through the IRB if you're on the Cornell campus, the Ithaca campus of Cornell, I should say. Has been historically limited access to the IRB application system, which can be a hurdle for investigators. The Englander Institute, I learned today, organizes a liquid biopsy biobank. So a biobank that is accumulating lots and lots of blood samples from cancer patients, and they have a universal consent of patients in place. And that's amazing. It's a great resource potentially for all researchers at Cornell that people can hopefully get access to. But then the question is, of course, why not all patients, or why stop at cancer? Why not develop these liquid biopsies more in other contexts? I have some affinity for solid organ transplantation, immune-related complications, and so on. There's also a question about healthy subjects. Often these studies require healthy controls, and each project seems to sort of try gather those samples and those resources independently. It would be better to sort of share these facilities. And then the last point that was brought up is to essentially replicate the efforts that have been put in place for the single-cell working group. It was briefly mentioned, I think, earlier today. There's a single-cell working group. Researchers here at Weill Cornell Medicine and in Ithaca often meet in virtual meetings where students present their latest research related to single-cell sequencing. And so given the interest on all campuses in liquid biopsies, there's maybe an opportunity to do that also for that particular topic. And it may be fun to also set up a listserv where people can bring up challenges, opportunities, questions, and so on. And that's all I have. So any further ideas or comments would be more than welcome. Yes, there.
Cellular vesicles happen to come up during your
Yeah, shame on us, they did not. No, indeed, yeah, there's exosomes and so on. There's a whole world of nucleic acids, things that float around in the blood that one can investigate and that are super interesting. And often we focus, the things that came up are the things that are in the immediate interest of the people in the room.
Hi. Well,
Yes.
I think it's important to specify is that only a fraction of that is coming from the tumor. You have circulating DNA, but only a fraction is coming from the tumor, and actually identifying that is not as trivial.
It's a good point, yeah. So indeed, and it depends a bit on the cancer type. It's been shown that for many cancer types, but certainly not all, that there's tumor-specific cell-free DNA present in the circulation. But often the levels are low, and it's all about finding the needle in the haystack. And so that requires, in a way, that's an opportunity because it's something that requires technology development. One big challenge actually is not just the sensitivity of these assays, but often specificity is an issue. As it turns out, there's a lot of biological noise, there's mutations that happen in our tissues all the time that lead to a background that can very much be a limiting factor for these type of assays. So, good point.
I want to make a comment about your point about the biobank. So Dean Choi has actually asked us to come up with a much larger biobank effort. It would be a blood biobank initially. There will be some tissue as well, but really it's going to be mostly a blood biobank. We want to make this biobank as useful as possible. I think to your point, I think if we can get suggestions about specific cohorts, specific disease, that will be of interest to the community, to the Cornell community in general. I think that will be very helpful to us. So if you have such ideas, please feel free to send us an email, with those ideas. This biobank has the potential to become very big. I mean, the numbers we have now are around 200,000 people enrolled in this potential biobank that the dean will help us fund. So we want to make it very helpful and include as many cohorts of interest as possible.
One of the problems we have actually is a space problem, and we've discussed that. And during another group session, we talked about that there is no space problem in the biobank in Ithaca. So perhaps we could do some collaboration with the biobank at Cornell Ithaca for the Eipm samples or something.
Yeah, that's a great idea. Why not also move the biobank to Ithaca? And so these shared biobanks are liquid biopsies are super responsive to this because, again, you can often do research based on archived old samples that have been previously stored, and that's really sort of a small logistical, practical aspect of liquid biopsies, but it makes a huge difference, right? You don't need to have access to freshly collected tissues, and so that makes it much easier to do larger scale studies that look at many more patients and so on, and to design studies. Yeah.
So one of the issues in cancer, which I guess applies to other, is just that there is a lot of commercial assays now available where you can do liquid biopsies, and more studies show that it does correlate with tumor tissue biopsies at the same time. So it seems to be validated that it's it's a good surrogate. But the other issue in terms of expanding and doing studies is just the sequential. Looking sequentially and to setting up prospective trials and et cetera. And I think more often than not, it's haphazard and not, clinically annotated as well as it could be. So I think that's one of the challenges that, I guess if you're expanding the biobank, you have to really figure out, and maybe it is just specific cohorts, but then you're going to do a study, and if you're going prospectively, it's going to be years before you're going to get your money's worth. So it's probably, I think it's problematic going prospectively, but that's what we have to do.
Yeah, it's a great point. It's also one that is, again, specific to cancer, I would say. There's other settings like transplants where these type of serial samples are more often available. Where the patient population is less heterogeneous, where all patients you go through the same surgery and then the same follow-up, where there's lots of clinical records available. So it's often specific to cancer, I would say. Yeah, that is all. Thank you very much.
Thank you.
All right. So moving on to work group 2, digital technologies for clinical care. I wanted first to thank all our collaborators for their valuable time and input on the discussions that we had. We were focusing on how we can bring more new digital technologies on improving our cancer care, our clinical care, and more specifically our cancer care, and essentially building on our precision medicine protocol. So on the IRB study that we already have, that we have a multi-omics approach, we want to add an additional layer that is essentially capturing sensor data from our patients, either passive data or active data, which is essentially patient-generated data from sensors or patient-reported outcomes. And we want to have that capability to track them during treatment and also post-treatment. So we discussed a couple of very interesting ideas, and we set some goals on how we can collect data and measure response to therapy. And we also wanted, as a second goal, to discuss how we can enable our clinicians to act upon that information and essentially provide a white glove treatment, or some ideas for, I think Bayan that was describing cachexia and how we could use BMI information, weight information to act upon it and give valuable information to the clinician. So in terms of action items, the group was originally comprised from Weill and Cornell Tech, but I want to welcome and bring other people from Qatar campus or from Cornell in Ithaca to join the conversation. And the action items that we want to start is to start with a Google Doc that we could start an ideate on the ideas better, and 2, identify champions and our clinicians that are interested. So this is going to be part of our pan-cancer study, and we want to find champions in each disease group to start building this together and essentially helping tailor to go to the acting upon data and potentially changing the light on therapy based on the sensor data. So I'll open up for questions. Yes.
As a clinician, I'm just curious, what are the digital technologies specifically you're thinking of that we would use, and how would we get it to the patient? I mean, just the whole process. I realize you're saying obviously you guys have a lot of different technologies. You're saying, okay, well, let's do it in prostate cancer, let's do it in breast cancer. But is there that discourse between us as the doctors and you as the technocrats to...
Then I'll sit down. I'll be a technocrat. Technologist. You were almost there.
Technologist. I'm close.
No, technologist. Okay. Yeah. Yeah. That's the conversation that has to happen. There are, and we're somewhat opportunists because there are things that we can get easily because they're already consumer technology that generates signals that we can interpret in ways that are relevant to you and your condition and your clinical questions, right? So you might want to measure for how many patients walk in as cancer patients a week or a month?
Hundreds.
Okay, so you might want all hundreds of your patients that are coming in every week or month to get, what's your field?
Let's say prostate cancer.
Okay, so now I'm riffing, right? But you might want to get some detailed information about after they leave and under treatment, what's going on in terms of urination and function and all those things, okay? Now we're going to be giving you some proxy, right? So anything that has to do with diurnal patterns, hours spent out of the house, time they left the house, how functional are they out there in the world, is an example. So anything having to do with everything having to do with sort of rate of walking speed and overall mobility, we can do that easily. We can do heart rate. We can do heart rate variability. We can do very detailed sleep monitoring. Some of these require buying them something versus something they already have. Patient-reported stuff is all around trivial. Like that, we can do anything. Function, performance status. Yes.
There was a study that was done looking at having an app for patients to do reported outcomes of getting chemotherapy and show that it improved survival.
Yes.
For instance.
Yes.
But you're saying you're going to the next level where you're going to sense, without the patient entering data, you're going to sense what's going on.
We'll do both, because I mean.
But that's not being done right now. So I haven't seen any studies, let's say in chemotherapy, patients receiving chemotherapy, where there's looking at, this telling you what their gait is.
I cannot guarantee you that it is not being done at all. I know we've been recommending it for 10 or 15 years, right? So I believe it's probably happening somewhere, but it's not being done widely. And yes, there's no reason not to get some basic performance or function or a proxy for a symptom level, particularly when it's not absolute, where you're hoping to see overall improvement and then you see a plateau or you see a relapse, right? And then we make it we can make it more specific to the nature of where the treatment is and what the symptoms or side effect or improved function that you're looking for. Recovery from surgery is different.
Are these devices available for now? You're thinking about the HRV and all these devices. Do you have them here so we can do them?
It would make no sense for us to have them here because anything we have, we would have purchased. They're not the latest, right? But for a small matter of some $100 per patient, right, you can buy various consumer items that we can create the plumbing to get the data from. Tanzim, do you want to say something?
No, I was just confused by what you mean have them here.
Did they have a battery of devices like actographs? Like in the old days of actograph, you would have a whole little inventory of them because the technology changed about every 5 years, and now it changes every 5 months. So we try to, by and large, sometimes we have people who design new, you want to do something brand new, you go to Tanzim and her students and they do something brand new.
So you do have
Hi,
Yeah.
Dr. Estrin. Hi, I'm Juliana. I work in Dr. Yo's research group. So we worked with you to develop we have an app to track patients after surgery where we use a Fitbit as a proxy for return to baseline in terms of movement. It's expensive and is only possible through a grant, but you can also use, for our controls, we're using the, I work in a group with Dr. Estrin that has an app to track return to baseline after surgery. And we use Fitbits because we're grant funded, but we also have a backup where we use the health kit data on iPhones and Android devices to kind of monitor return to baseline and their steps and their movement and that kind of thing. So it's totally possible. And then we actively ask patients to take photos of their incision sites, their urine, to report their temperature, report various, to report various clinical indicators daily, and it alerts physicians. So this is possible and happening here right now.
And one thing I just want to mention on whether you have those devices here or not. So let's say Apple Watch now can do EKG, right? So having a bunch of devices often doesn't even make sense. You want to either utilize the devices they have or whatever the next generation of devices, and rather than having something that's from 3 years ago. So yes, we have a bunch of heart rate variability devices and chest straps, but yeah. Here, with me?
Yeah.
Yes, yeah.
But
I wouldn't make a big,
But I actually think it's the wrong question.
yeah.
Because anything that somebody has that's not used is probably
Old.
This stuff is too inexpensive compared to everything else you do. That I think it's about what device might your participants have, what might you want to get for them, compared to the cost of doing the plumbing and getting the data and processing it.
If you have them, we can basically borrow them, I guess, for free, because they're old, you know? They can do the job, you know?
I'm worried you'll be wasting your time. I don't want you to waste your time.
Because
You generate a lot of data that isn't as good as the newer things.
exactly, and whether the patients, if they're wearing a watch that will give you data, you're going to get better compliance, continuous data, than asking them to wear an old device. So
So let's talk it through, but yeah. Okay.
But isn't the issue, I mean, just to chime in on this, I mean, isn't the issue that, every patient has a different device, and how do you do that? How do you, I don't have an Apple Watch, just, so if I want to enroll in a, what do I do?
It's not as variable as their metabolomics. I mean, it's like, yes, it's not everyone has the same, but it's so much less variability than all the other stuff that you guys deal with every day.
Okay.
And then we do a little convenience sampling initially.
Sure, you're confusing the thing. There are two different things. Of course, metabolics are different, but HRV is also different. So, what Olivier is saying is something completely different. You should have a standardized tool to measure those. We do have standardized tools for metabolomics. So, it's just, I'm just saying,
But you get steps from iPhone.
I got your point, by the way.
You get steps from iPhone, you get steps from Android phone. They're not identical.
And they don't need to be
They
identical.
don't need to be identical.
Heart rate variability may be different, but steps don't need to be. So we have to get into the details.
Heart rate variability is very different.
But I mean, what we found is that you're mostly tracking trends for each patient, right? So like you're looking at what happens to them and how they use their phone, and it's very individualized. And in terms of Dr. Elemento's question about having different devices, you get a really good developer who allows you to extract data from different phones and different apps, and that's how you do it. It takes more money to set up the plumbing, like excuse me, Dr. Estrin said. I'm sick, sorry, but it is feasible.
And just to tap on that point, I think working together between campuses, there is a lot of domain expertise that we can tackle together. The key idea is to enable clinicians like Dr. Nanas, Dr. Sternberg to have additional insights in their arsenal when they're evaluating their patient and what's the best line of therapy. I know we have to move on to the next group, but I would invite everyone that wants to continue the conversation to follow up with us later on so that we could discuss and make this an active, contributing effort. Thank you.
No, it's okay. notes. Hi, everyone, again. I will summarize the engineering ex vivo models group. First, also I want to thank all the participants for a lively discussion about organoids. I think we agree and disagree on many issues, but in summary, so it came the question on what do we use, right, for our investigation? 2D established cell lines, organoids, PDXs, and we all agree at the end that it all depends on your specific question. They are all models. They all have advantages and disadvantages, and so, but the field is moving towards more scientists are using more and more 3D organoids. And that's being reflected by an international consortium that's being formed, funded by the NCI, that will release, they aim to release 1,000 3D models through ATCC to be available to the broad scientific community. So that will facilitate a lot research done on these models to answer the questions that we were asking today. How well they represent the tumor, compared to these cell lines, how well are we representing the heterogeneity of the tumor, right? Are really a much better model as we claim they be. So there is anecdotal evidence about that, but we still need more data. So the next question was, do we exploit the models as we have them now, or we develop the technology to the next version, right? Version 2.0, to probably 10.0 and beyond. And I think we decided on doing both, actually. So we are close to have our high throughput drug screening platform ready to use. And I think that's a very nice platform where everyone can come together and develop drugs on the models that we currently have, that everyone has. But then also, we need to make efforts to develop the technology further, right? So we know that a tumor organoid, it's still only the tumor cells, the epithelial cells of the tumor, and we are missing parts of the microenvironment. And we discussed the extracellular matrix. Right now, the standard is to use Matrigel as the 3D support, but we should move towards a more specific matrix that reflects better the matrix of the tumor, and then also other parts of the microenvironment like the immune cells. I think that will be probably the next step, is developing immune-competent organoids, and then also other parts of the microenvironment like stroma cells, fibroblasts, and even the normal cells that are surround the tumors. And then also the use of systemic factors, so using a matched patient serum that for drug screening specifically that might affect drug response and growth of the tumor. Then we discussed the use of technologies to characterize better the current technologies, right? Like right now we use genomics, transcriptomics, and we discussed about using proteomics, metabolomics, that they are not fully used in this field. And also single-cell RNA-seq. We were discussing if that would be a better tool to understand tumor heterogeneity and representation in the organoids. And also a concept of more than genomic or transcriptomic or even proteomic state of the cells. It came up by Howard Fine, actually. He proposed the concept of the cell state, right? Because maybe you are saying the cells are not representing very well the tumor, but actually they are converged towards using the same pathways that actually you can target with the same drugs. And then another issue was, okay, more and more people are using organoids, but still a very expensive technology. And so, yes, the answer is yes, it's very expensive, but I think the more and more people will use it, it will become less expensive, and also changing from matrigel to artificial matrices, and also trying to work together towards a miniaturization of the technology. Probably will get the technology even cheaper. And the other discussion was there's a lot of research. We claim that these models recapitulate the response to drugs in patients. That's what we want. That's why we use them, right? To mimic the tumors. And how do we do that? How do we make that step further to like the patient to organoid, organoid to patient? And I think that's where we are going. In part, having the CLIA certification in our high throughput platform will make that easier. But if you have any discussion on how we could start doing that, it's very welcome. And another topic was, so there is data already published about drug screenings, how organoids recapitulate the tumor response. But at some point, I think the idea is that we build a data repository, a library of information about organoid, genomic, transcriptomic, all the characterization of the organoid, that at some point, when that 1,001 patient comes, we don't need to grow an organoid because we will know, based on all the characteristics, how it will respond to certain drugs, based on all the functional assays that we are doing now. And so that the process that we are currently doing will inform the decision of the treatment to patients. And these, of course, all these efforts and challenges in the technology are not, will not be obtained through one single lab. This takes a village to do it. So I think as everyone was trying to make a focus on making a collaboration tri-campus, right? The expertise of each of the campuses and come together to try to move the technology forward. If you have any question. Okay. Okay.
Thank you.
Thank you.
Just quickly, if anyone found a black backpack that has Brian on the lanyard, can you just double check to make sure that you have the right bags? So we misplaced it. If you see it around. Okay, yep.
Great. That was surprisingly easy. So Cora, Erica, and I led the stakeholder engagement session, engagement and education in precision medicine. And we had a diverse group of folks who were there across campuses, across educational spectra in their careers. And I think we all acknowledge that this was a tough topic. There's a lot of stuff going on and it's complicated. And so this is kind of what our bottom line was here. So we looked at engagement from two perspectives. One from the end user, and the other one from the healthcare system. And so from the end user perspective, we thought that specifically engagement and education with patients needed to involve a couple things. And so one was the use of technology, such as mobile phones and social media. We talked about directed marketing and messaging for patients, leveraging some of the existing resources we have and the existing expertise across the institution. Talked about leveraging patients' trusted resources. And so that means a couple things. You know, patients trust their primary care providers, who often are not the ones who are suggesting involvement in precision medicine. And so that was one avenue. They also are influenced by their own communities, and communities are very, very influential in patient behavior. Other thing, and I think this has been mentioned before, was the measurement of patient-important and patient-reported outcomes. You know, what's important to us as researchers and clinicians and academics isn't necessarily what's important to patients, and so ensuring that there is uniformity and synergy there is important. From the physician and healthcare system perspective, again, this is hard. You know, if precision medicine implementation was easy, everyone would be doing it. It really has to do with the healthcare system that is that you're talking about, whether it's an integrated healthcare system, whether it's an academic center, whether it's a private model. We talked about overcoming provider biases in terms of how they view patients, in terms of patient recruitment to trials, as well as knowledge gaps. You know, this is stuff that most people have not learned about in med school, and that has implications as to how you practice. And then the last thing we mentioned was, you know, I think everyone sees the arrival of population-based, population-level precision medicine. And we strongly felt that it mandated stakeholder engagement. And specifically, we looked to the PCORI model for stakeholder engagement. And so for those familiar or not, PCORI is the Patient-Centered Outcomes Research Institute. It was brought into existence when the ACA came into effect in around 2010, and is meant to specifically fund comparative effectiveness research that involves stakeholders, that brings together multiple people with the idea that you create research that endures by having all the end users, all the stakeholders at the table. And so this is a model that at least we felt in the large integrated systems that have been able to successfully implement precision medicine on a population level, they've brought in patients and purchasers and payers and industry and all these folks such that everything is thought out from the get-go. Also, from a research perspective, it's a slight bend of the road from what NIH usually advocates us to do, but we have a pragmatic trial going on that employs a similar framework, and I think the end result hopefully will be more durable as a result. So yeah, that was the summary of our discussion and our findings. Questions or concerns or comments?
I just wanted to make a comment. We were discussing how we're going to reach our audience and our broad audience, and the broad audience often then goes to their internists. And we were told that at Cornell Ithaca that they have incredible IT, business communication people, marketing people, and that one of the physicians, the head of the biobank there, had actually paid for an intern to go work there, work with them, and understand how to do the best kind of business and PR so that they could reach out to the community. Because I think that's one thing we haven't been doing well, because we're not businessmen, we're not marketers, we don't really know how to do that. And perhaps we could use the people in Cornell Ithaca to help us. We could get someone to go there and explain to them what we need, or whatever. I don't know, someone could do an internship up there and then come back here, and then or use their knowledge, because they have a whole experienced team of people who know about marketing and communications.
So I'm not sure I know what question you're addressing with your answers here. Is the problem more that people are unaware that these personalized treatment options exist, or is it that they are aware but they're saying no to the alternative?
I think every I think every problem exists. I mean, there's problems regarding awareness on a patient as well as a provider level. There's problems regarding access. I mean, there's every disparity in healthcare is magnified in academics and in research, and particularly here. So it's broad.
Maybe I would just make one more pitch for some of the resources that occur up in Cornell Ithaca. Some of you might know of the Physical Sciences of Oncology Center, which exists between the two campuses, and which various investigators related to Meyer Cancer Center down here are involved with. But the Ithaca half of it is very actively involved in a local outreach community to cancer patients, and meets directly between researchers and the patients, and not their clinical providers, but the actual on-campus researchers. And so they've taken a lot of endeavors to provide basic understanding to the patient population that has really, I think, a model system. So we could certainly help put people in contact with the leaders there. It's a very student-led initiative, and it's, I think, beneficial to the researchers to see the impact. And we're so much more removed than people are here, so it could be another system to look at.
That's great. That's a great idea.
Right. So that's the end of the working groups, the kind of discussions and summaries. We do have one more panel session left that's focused on cross-campus students and trainees, kind of meeting other mentors. But if everyone wants to take a break and come back, or we can jump into the next session.